there we go. All right, so the laptop's recording. And now I'll just post the lecture as a presentation. Okay. Paul says bottom bar. Yeah, thanks, Paul. Sorry, got it kind of slow. So today's lecture, I'm going over the food pathogens. And in this first picture, I've got a little tiny message from Health Canada in the top corner. There's this little cartoon. It says, how do you treat leftovers? And it gives you a time of how long you can leave out things like cooked meat, stew, eggs. How long can you leave out fish and poultry, gravy? And I know it says here for gravy, you can only leave it in the fridge for 3 to 4 days. My goodness. I'm bad at this. Sometimes I have gravy in the fridge for a week or two before I use it up. Yeesh, don't tell my wife. So this image here shows a person taking care of some soil. What they're doing is they're autoclaving some soil and vegetable sort of dried twigs and grass samples, and they're putting it into an autoclave. The autoclaves can be fairly large, and this one, as you can see, is huge compared to the autoclave that we have in our lab. Okay, here's just a review slide. Last time before reading week, so many weeks ago, we were talking about the germ theory of disease. I talked about what is germ theory, who's Louis Pasteur and who's Robert Koch. And I talked a little bit about typhoid Mary. Okay, so here's a review question. And I saw Dawn was online and Neil's online. So Dawn, Lindsay, Amy, Neil, Case, and Cassandra, this question's for you guys. Oh, Paul asked, is that a mushroom substrate? Could be. They do make sterilized mushroom substrate out at very large autoclaves. Okay, but first question for Dawn, Lindsay, Amy, Neil, Case, and Cassandra. What is germ theory? Remember germ theory, looking for the definition of what it is. And it's very simply stated. Germ theory is crickets. Germs can lead to disease. Neil says that. Yes, Neil, correct. Thank you. And when we say germs, we mean the microbes that cause disease. That this is something that Pasteur proposed, and we've since many a time confirmed it. How does the germ theory differ from miasmatic air? So what is miasmatic air? If you remember that, it's bad air. How is that different from bad air and spontaneous generation? Paul says, can I advance the slides? I thought, can you see slide 3, Paul? I'm still on slide 1. Oh. Well, that's strange. On my computer, I see slide 3. So let me just try this instead. Oh, that worked. Okay. Question 2, what is the boiled beef experiment by Louis Pasteur? If I switch PowerPoint to presentation view, it should go bigger for us, but when I switch to presentation view, unfortunately, you couldn't see it. It must be a bug in Zoom. So if I can't, how's that? Boiled beef experiment. Don, Lindsay, Amy, Cass, here's a question for you. What was the boiled beef experiment by Pasteur? Remember, this had to do with 3 vessels, 1 vessel being opened, 1 vessel being sealed, and 1 vessel with a S swan-shaped neck. Cass says, was that bad air? Miasmatic air. Yes, Cass, that's correct. With the boiled beef experiment, we have 1 vessel that's open. If you leave that open to the environment with some broth in it, microbes come from the air, and then eventually it spoils. If you have a vessel that's sealed off at the top, the microbes from the air don't land in the vessel, and you don't get a spoiled broth. And finally, if you have a vessel that has a swan neck, a long swan neck, the microbes can fall at the bottom of the swan neck, but they don't travel up the swan neck inside the broth to spoil the broth. So this proved that if you have broth open to the air, but in a path that microbes cannot take, it will not spoil the broth. So this also disproved spontaneous generation. I'm going to say list Koch's postulates next. What sickness was described to explain Koch's postulates? Can you see that? I'm waiting for people online. So that's Don, Lindsay, Amy, Case. Cass, I got your response. Neil, I got your response. So I'm just waiting for the 4 other people.
Sweet.
There's 4 postulates. First postulate, okay, maybe I'll just go over them. First postulate, you isolate bacteria from an organism that was killed by disease. Second postulate, you
Put it into a living host.
You obtain a pure culture of that bacteria. And then the third postulate, you take that bacteria, infect another organism. And then if that organism infects and kills the host, it shows, and then what you do is you re-extract that dead animal to show it's there. I can hear somebody. Somebody's got a live mic. And I know I can control the mic, but I don't see it up past. Could you turn off your microphone? That's okay. Okay, so if somebody could type out a contribution from Louis Pasteur or Robert Koch, that'd be appreciated. Thanks, Jackie. We've got some background noise from people in the hallway. Jackie's taking care of that. Contributions to public health, people. What you got? Thanks. I don't know if you heard Amanda. One of the contributions from Louis Pasteur is pasteurization, sanitization methods. In the case of Robert Koch, sanitization of clothes and bed sheets from hospitals to prevent disease. That's one of the contributions from him, because he realized that saliva can contain dried tuberculosis, and the dried tuberculosis is contagious. Okay, so that's review questions from lecture 6. Let's carry on to slide 4. Slide 4, this is my overview of what I wanted to cover today. I wanted to go, if there was more people here, give you guys the Typhoid Mary video, but I think we'll skip that. But I do want to talk a little bit about the idea of food infection and toxic infection from food one more time. And what I'm going to do is talk about 2 serious Canadian case studies, as well as Clostridium, botulinum, as diseases that can occur in food. So I'll talk about what happened at Maple Leaf Foods. I'll talk about Clostridium, botulinum, and I'll talk about Walkerton today. Parts of this lecture, I might just go over very, very quickly. For example, the part that was viruses, mold, parasites, prions, that part of this lecture you've already covered in an assignment for me. So that was done before break. I've removed that from the lecture totally, and then we'll talk a little bit about sterilization at the end. So food illness by microbes. If you have an illness by a microbe, that's referred to as a, I'm just going to move this out of the way. I don't see the whole slide. I want to get rid of the bar on the left, normal slide sorter. There we go. Any illness that's caused by ingesting food contaminated by microbes or toxins is foodborne illness. So it's a very general kind of definition. And we consider 2 kinds of illnesses. Like if you take the bacteria, ingest it, there's an infection that leads to sickness, that's one kind. If you take the bacteria, and the bacteria can be dead and not living at all, but their toxins are still there. If you do that, and then this is the case for Staph aureus and Clostridium botulinum, you can take in the toxins. And if you take in the toxins, that's what gets you sick. And so I'll talk about E. coli and botulinum as being caused by toxins, and I'll talk about Listeria, which causes an infection that is caused by the bacteria. Okay, so like I was saying earlier, You can be sick from bacteria, viruses, mold, fungus, parasites, or prions. All of those are different ways of getting sick from foods. Now, there's this idea of foodborne intoxication. That's a specific case of food infection. In this case, bacteria toxins that the microbes make, they're a byproduct of the organism being alive. Oh, Brooklyn says, could I go back? But I don't know where. Back one slide, please. To the foodborne biological hazards, Brooklyn, is that what you're talking about? And do you have a question here? Biological hazards cause spoilage or pathogenic microorganisms? All these slides are posted. If you want, you could download them and follow with that, as well as the online lecture. Toby, what's that? Don't worry. Okay. Okay. So I'll carry on. As I was saying with the foodborne intoxication, you can't deactivate the toxin just by cooking it. For example, if you just cook Clostridium botulinum toxin, you're not going to deactivate it. There's other ways to deactivate it. And what happens is you take in the toxin, and that's called foodborne intoxication. So can't deactivate them just by cooking them. So if you cook a food that has Clostridium botulinum, you'll kill off the bacteria, but you won't get rid of the toxins. And that's the case with the E. coli infection. If you have E. coli in your food, whether it's raw food or vegetables or untreated water, you can cook that, but it has the toxins in it. And it's the toxins that make you sick. It's not the bacteria themselves. So you'll kill off the bacteria, but you'll still have the toxins. It's the same thing with B. cereus. You can have rice, leave it on the counter overnight, heat it up again in the morning. You could get sick from that because it might have the B. cereus bacteria in it that has toxins. There's E. coli, there's Staph aureus, and Clostridium botulinum, as well as, those are the 4 that I wanted to mention. Okay, so I've covered this before. Yes?
The toxins. What is the quantity of the tolerance that needle?
What is the, Tovechku's question is, what is the point?
Of the toxin that is produced by the pathogens. That it is that later, like if it's not in the food.
So what is it exactly that is a toxin is kind of, I think, what your question is.
So that toxins are chemicals that are produced.
They're not chemicals that are produced. Typically with bacteria, they produce proteins, and these proteins are what are the toxins. So if you think of a protein as a chemical, we don't think of them as chemicals normally. We think of proteins as being a biological molecule that an organism makes. And it's these proteins that are toxic. That would be good. I could show you a picture of a bacterial toxin,
Okay.
maybe next time.
I know, but you know that like protein it's not as if it's biological, but it's made up of atoms and molecules. So you can say it's also like chemicals, but it's higher up on that chemical because they are made up of so many atoms and molecules. It's still not the living thing.
So Tabachku's question involves toxins, and he's asking, so there's proteins are made of these chemicals. How is it that these chemicals can cause such an effect? And I have a slide later with botulism, so I'll show you that in a bit. Okay, so like I was saying, there's foodborne infections. These bacteria, if you have these bacteria in your food, they can make you sick. So Listeria monocytogenes, Salmonella, Vibrio, cholera, Campylobacter jejuni. This is in chicken, often Clostridium perfringens. This is in beef and gravy, chicken as well, and Shigella. So all of these, if you have the live bacteria in your food and you process food, you can pass the live bacteria to other people because it's in the food. And symptoms of infection: nausea, vomiting, cramps, chills, all kinds of symptoms from this, okay? I'm just gonna skip ahead to the questions. So there's the foodborne infection and foodborne intoxication. Don, Lindsay, Amy, Case, I still haven't heard from you four. Do you have difference for me? What is the difference between foodborne infection and foodborne intoxication? Give them a minute to type out something. And so foodborne intoxication could include infection like a Clostridium, botulinum infection, E. coli infection. Foodborne infection could be a Listeria infection, where a Listeria monocytogenes infection. I haven't heard from anybody. Do I give more time? Okay, so the foodborne infection. This is a general case for both the foodborne intoxication as well as what happens if you get, actually the foodborne illness is what you get in both cases. Foodborne infection is what you get when you have the bacteria ingested and the bacteria multiply and you get sick from that. The foodborne intoxication, you ingest the toxins from bacteria or yeast or whatever microbe you have, and you get sick from the toxins. And common symptoms. Anybody want to give me a couple of symptoms? Chills, diarrhea, nausea, all that sort of stuff. Those are all symptoms of foodborne infection. Okay, so next slide. I think I talked about Salmonella, and I talked about this before reading week. So Salmonella poisoning can come from anything like livestock, including hogs, cattle, poultry, birds, or any pets you might have, reptiles or insects. So it can come from animals, and there's two strains. There's the Salmonella enterica typhi. This is the agent responsible for typhus. The disease known as typhus. And then there's Salmonella species, which is if you get this general species, it can be referred to as Salmonellosis. So here's a picture. I shared this with you guys just before break. Someone's got a plate with selective plate showing how to grow Salmonella on a XLD agar plate. It's a gram-negative rod. It survives as a facultative anaerobe, meaning you don't need oxygen to allow this thing to live. And it has this problem where it can survive in refrigerated or frozen foods for a long period of time. So if you freeze it and then thaw it, you can still have the Salmonella there. If you refrigerate it, it doesn't kill the bacteria. But if you heat it, you can kill it by heat. What are symptoms of Salmonellosis? Salmonellosis includes, again, fever, diarrhea, stomach cramps, nausea, very similar to other foodborne illnesses, but it can be fatal. So you can be sick from this bacteria within 6 hours or 3 days. So it can take some time for this infection to take place. And if you're very, very young, like as a baby or an elderly person with a weaker immune system, then it's very, very, you're susceptible to it. And then there's a point that the bacteria seem to survive within the gallbladder. So it's show you it's right inside of the gallbladder, as well as the urinary tract and the brain. So it can affect different parts of your body. And I talked about this where it's the outer shell of eggs that can get contaminated with fecal material for Salmonella species. So it's not just typhi, but also the other Salmonella. I'm getting both of them kind of confused here. But one of the most famous examples, and I wanted to share the video with you. Again, it's on YouTube. Go ahead and watch it. This is kind of a sad video of the life of Mary Mallon. And Mary Mallon was this person who was a cook. She would work at one house. People would die. She would go move on to another house. She would cook. People would die. She would lose her job. She'd go to another house. And every place she went, she was a cook. And she took care of the people who were sick, but still people would get sick. And they didn't realize that she was the person carrying the disease until they tracked her down. So this is like the very first example of tracking for a disease. And they tracked it to one individual, and she had no symptoms. So this is where we get the idea that you can be an asymptomatic carrier. Okay, so we have this new term from Mary Mallon's existence, and we studied her that she had the disease, but she had no symptoms. So she's asymptomatic. And Robert says Typhoid Mary is a cover. It's the CIA that did it. Sadly, Mary Mallon did suffer for this. She was brought to trial, told not to cook anymore. She went to housekeeping, and then she couldn't make ends meet, so she went back to cooking again. More people died, and then they realized she was cooking again, and then she spent the rest of her life institutionalized. And this is what can happen if a government uses a heavy hand to like isolate somebody. And it has happened in the United States. It's kind of a sad story.
There are drugs that kill the Salmonella bacteria. Why did they give that to like get one?
good question. Tavachku is asking, why didn't they have drugs to kill the bacteria? That antibiotics that weren't invented till the 1940s and 50s. And she lived in the 1910s, 1920s. So they weren't available to her at the time. How to prevent salmonella poisoning? Avoid direct handling by infected employees. So if you know you're infected, don't let that person in the kitchen. If you acidify your food, so if you add a little bit of vinegar, that'll help kill off any salmonella. Wash fruits and vegetables. It's one way of removing the bacteria, but they can still be inside. So it's not 100% effective. But if you heat the food, that kills the bacteria. And I have here growth inhibited by 0.1% acetic acid. If you use sanitizers that are used in the food industry, they'll kill the bacteria as well. Also, if you pasteurize your food, that'll kill off the bacteria as well. So there's ways we can prevent salmonella poisoning. There's a slide about salmonella regulation changes in Canada. And here's a picture of Mary Mallon, photograph from the 1920s. And I even linked the Guardian article I got this from. So you can see she's photographed laying in a hospital bed in isolation in New York. And here's a question for Don, Lindsay, Amy, or Case. Still waiting to hear from you for, I don't know. Don is here. Lindsay not in. Amy not in. Case. Cass has already answered the question. Case not in. Okay, so Don, haven't heard from you yet. Can you tell us what a asymptomatic carrier is? Two words for the definition. First word is part of the, why is the letter A used at the beginning? It means no. And then the second word, symptomatic. So no symptoms. Don? I don't know if she hears. Okay. Asymptomatic means no symptoms. Okay. I'll carry on. Oops, I'm going the wrong way. Sorry. I'm going to skip the slide and carry on with the story about Maple Leaf Foods. Okay, so what happened at Maple Leaf Foods? Back in 2008, there was a very, very serious contamination issue at Maple Leaf Foods, dealing with this bacteria called Listeria monocytogenes. What is this bacteria? This bacteria is found in dirt. It's found in sewage, water, silage. It can be found anywhere there's like animals bringing up dirt to the feed or whatever. And it can be found in cattle. It can be found in sheep, chickens. It can be found in milk, raw milk, that is. It can be found in pooled water, as well as they've isolated it from worn tiles, ventilation systems. So this sort of bacteria seems to be an environmental bacteria that can be found in a lot of different places. Again, this bacteria is what I refer to as a facultative anaerobe. Facultative means it can survive in both, but normally it's anaerobic, so it doesn't need oxygen, but it'll survive in oxygen. It's called a gram-positive, so if you're to do a gram stain, it'll turn blue. It doesn't have spores, and here's what makes it so bad. If you heat it and thaw it, it can still survive. If you dry this bacteria down to like absolutely dried state and let it sit there for years, it can still come back. Also, if you refrigerate it, it's fine. It survives in refrigerated conditions. So, and it's also tolerant to 10% salt. So if you have something that's very salty, Listeria monocytogenes can survive there. If you have it heated, thawed, dried, refrigerated, it can live there. So if you think moist, cold environment, what kind of place is that? If you've ever been in a meat factory, I've never been there. My wife talks about it all the time as this cold, damp place. And this is what a meat factory is like. Tovechku's, sure, go ahead, what? What kind of meat did you have to process there?
When they make this sausage used by this Tim Hortons and stuff like that, the round one.
Oh yeah.
Where it's milled and then shipped into the round.
Great. So Tovechku's sharing
Cisco.
that he worked at Cisco making sausages for Tim Hortons. And people make sausages at home. I've been around friends, family that make sausage at home. They just put the ingredients into a meat grinder, push it through, and grind it all up, push it through a tube, and they have the animal casings, and they just take the animal casings, put it. Yeah, so it is a very common sort of thing, whether you work in a factory or do it at home, you can do this. So monocytogenes, what is the risk? Where can you find it? Raw milk, cheese, ice cream, because ice cream comes from milk, raw vegetables, raw fermented meat sausages. So sometimes meat sausages are cured, they're not cooked. And cured, by curing, I mean they're exposed to high levels of salt and allowed to stay at room temperature to cure. Raw meats, all kinds of raw meats can have this bacteria, smoked fish, as well as ready to eat foods that have been contaminated after cooking. So lots of different ways to get this bacteria infection. So, so we just keep things clean. Keep things clean, sanitized. You can reduce the amount of this threat. What are the symptoms if you have an infection? Again, vomiting, nausea, diarrhea, fever. It's like a flu. And if you have a baby, it could be loss of appetite. They could be kind of lazy. The skin could go yellow. They could start vomiting. And Dawn says, sorry, she's she missed the question. Okay, we'll save another question for you up ahead, Dawn. Hopefully it works out. We're talking about the symptoms here. There's a documentary. It's only about 3 or 4 minutes if you want to watch this. It's available for you to watch online. And what it can do is if you have this infection, is it can affect the central nervous system. What happens is the bacteria get into the nervous system and causes meningitis. And meningitis experiences a severe headache, and it can cause brain swelling, and this can be fatal. So it's a very, very serious condition, and it requires a really serious medical treatment. I've met one person who had I don't know if she had a Listeria infection, but she had meningitis. And you can have meningitis that's viral or bacterial. And this person had gone to hospital for a week and had undergone an extremely invasive sort of antibiotic treatment. It's very, very hard on a person. If you have the Listeria monocytogenes infection, it can also affect a baby's placenta in a pregnant woman, and that could cause a miscarriage. How do you kill it? Asks Brooklyn. What we do is we keep the areas that we clean or prepare our food clean, and we also use ingredients that are pasteurized. There's two ways to do it. We'll see some other ways. Okay, so oh, here it is, prevention. How do you prevent getting sick? So if you lower the pH of your food to below 4.4, acidify your food somehow, lemon, lime, vinegar. If you heat your food above 70 degrees Celsius, so you know how they say chicken is done if it reaches a certain temperature, chicken, turkey, beef, cook it to a well done temperature. Inactivate in vegetables by lysozyme. I don't know why this is there. Lysozyme is not a standard sort of way of preventing it unless there's, anyway. Sanitizers are much more common, and sanitizers can include some sort of alcohol cleaner, quaternary ammonium compounds. Quaternary ammonium compounds is being used by everybody in this class in lab. When we go into the lab, I ask everybody to clean off their desk with Quattro. This is a product that comes from Quebec. It's in the red bottles. That's an ammonium quaternary ammonium compound that's in there, and that's a disinfectant. So if you use a sanitizer like that, we've decontaminated the food surface or work surface. Okay, so what happened in 2008 at Maple Leaf Foods? So the case of the food infection is studied by a lot of people. It's even studied in legal circles because this is one case where a company had a real devastating effect, but the response of the company is to take responsibility, and they've paid the price, and they've tried to improve the product and improve their services as a result of what had happened. And you'll see here what we have. So at Maple Leaf Foods in North York, I got lines 8 and 9 being which lines it actually came out of. There is over 200 contaminated products, and everything that people got in their hands at home with the 97B could have been possibly contaminated from Maple Leaf Foods. And the bacteria are in the deli meats. So usually deli meats are cooked and they don't have pathogens, but they think that the contamination happened during the packaging of the meats. you're here. Good to see you. The outbreak at Maple Leaf Foods occurred in July, and what happened was people in the community were reporting food infections, and it started getting more and more. And they think that one of the problems was the federal inspectors didn't spend enough time at the food plants. Sometimes they don't spend more than 70 minutes in a plant. So they come into the plant, they do their inspection, they go to the next plant. So this may have contributed to that as well. And it said here it spent less than 5 hours a day, and sometimes as little as 70 minutes. Okay, and this is the response from Maple Leaf Foods. They had a voluntary recall of the food products. And they they identified the Barter Road factory. That was the place it came from. I'm just gonna move this aside so I can see. And there's a quote by the president of Maple Leaf Foods. Tragically, the products have been linked to illness and loss of life. Those people who are ill and the families who lost loved ones are offer deepest, sincere sympathies. Words cannot begin to express sadness for their pain. And I think over 20 people lost their lives from the food infection. And this is kind of like a repeat of some of the information. So they found it on lines 8 and 9 at the North York facility. And the plant went under intense cleaning, and they used sanitizers, including peroxyacetic acid, quaternary ammonium, isopropyl alcohol, and refrigerated gel, and a granular compound. So they disinfected and cleaned all of the equipment, all of the packaging equipment, to make sure it was all sterile and clean. 600 people had to attend training sessions on how to get rid of Listeria, and 250 people were laid off and while the plant was being cleaned. So you can see they've put a lot of effort into trying to get rid of the problem. It says here the cost was 20 million, 10 times the original estimate. So they thought it would only be 2 million as a cost, but I think due to the settlements as well as the cleaning, it's quite expensive. Okay, so what makes Listeria monocytogenes difficult to get rid of?
Surviving extreme physical conditions and chemical conditions, like the cold, in cold, and in substances with low concentration, like salt concentration.
Yeah, if it's at low salt concentration, Tibetka's saying, it can survive. It needs high heat to kill it off. It has to be over 70 degrees Celsius. Otherwise, it could survive. Natalie?
Just to touch on one of the things with regards to surviving the heat of cooking off, I was listening as I was driving in, and we were talking about was it botulism, or one of the bacteria surviving, like if you cook it, you kill off the bacteria, but not the toxins.
Yes.
And so how then those toxins are still,
How do you get rid of the toxins?
yeah, like they can still make you sick.
Yes, the toxins can still make you sick, even though you've cooked them.
So how would you react? How are those reactivated? Is what it is. Because if you,
This is the case with Escherichia coli or E. coli, as well as Clostridium botulinum. Both bacteria have this toxin if you heat the material, the toxin's still there. So how do you get rid of it? You have to just not eat it.
Because they recommend use like with meat, for example.
Oh, you may acidify too. I think that might help with the denaturing of the proteins.
It can denature the proteins, but can you eat it? If you acidify it up to the extent that if you acidify
Yeah.
up to the extent that you can't eat it for it to denature the protein.
That's a, when I say denature the protein, I'm thinking egg white, you heat it up, it turns white. So the proteins have been broken down. I'm wondering if
but some proteins are not denatured like that. Like
not as easily, correct,
Yeah.
yeah, they're not easily denatured. And this is a question that's difficult to answer. I have some points, but it's not a thorough sort of explanation. What makes Listeria monocytogenes difficult to remove? Difficult to remove. It can survive salt, low temp, and there's another survival condition, low heating. Yeah, it can survive up to 70 degrees. If you can get it above 70 degrees Celsius, you'll kill off this bacteria. Second question, what are some sources of Listeria monocytogenes? And I have, there are several environmental sources. In the 2008 Listeria monocytogenes case, what was the suspected source of contamination? It was the packaging lines. It wasn't the meat itself. It was the packaging equipment that they think was contaminated.
what packaging?
They didn't clean it enough. So they might have exposed the packaging equipment to meat that was contaminated, and then they cleaned it, but it didn't clean it enough, probably. And last question, how is the equipment at the plant disinfected? Various disinfectants were used. And the disinfectants included alcohols, quaternary ammonia. They don't mention bleach.
Some of this,
I think bleach is a little bit basic for some.
And it's too extreme, yeah.
like the food industry, so many of these companies don't use disinfectants. They use high pressure water.
Oh, yeah. And is it high pressure steam water that they use?
The water is a little bit hot, but not steam.
Not hot,
It's a little bit hot.
a little hot, okay.
But not steam. But there isn't to clean them as much. I don't see them.
Do they put any chemicals in the water at all?
It's on the tap water. It's on the
Tap water, huh?
It's on the line, the hot water line. That's not too hot. It's not steam.
So high pressure to remove all of the
Oh, that's not
meat.
something.
And
Although it should be able to remove the bacteria.
if it's like why we use a toothbrush in the morning, we use a brush to clean off our teeth, right? So we try and reduce the number of bacteria on our teeth with the toothbrush. So in your case, you use the high pressure water to just push off the bacteria from all the cleaning, all the food preparation material. Yeah.
Because there's a
So we're having a conversation here. Tavechku is talking about cleaning at a meat factory, and he's got some experience. It,
I think it still does not remove what the bacteria is possible. The high pressure,
it doesn't kill off the bacteria chemically, but it may remove it by abrasion, like just by knocking it off the surface.
Yeah.
And letting it go down the drain. Right? Okay, so that's the review of the Listeria infection. Here's Clostridium botulinum slides. Just a warning to everybody, there's one disturbing image of a drug user that I share as a picture to just, and Paul's got a link to Maple Leaf Foods, how they identified. I'll have to look at that. Thanks, Paul. So Clostridium, if you look at it under a microscope, it has a rod shape, but it's also kind of a club shape. This is a gram-positive bacteria. It's found in environmentally dust, soil, mud, as well as fish, shellfish, vegetables, honey, uncooked vegetables, grains, feces, carcasses of birds, animals. So it's an environmental microbe. It's found in a lot of places. And when I say it's widespread, yes, you can find this in a lot of different places. I have here that there's a Canada-wide food recall for President's Choice baby food in 2017. So that's only a few years ago, 5 years ago, they had a recall because there was some concern with Clostridium botulinum. If you have food cans at home that look swollen, my suggestion is don't even open them, throw them away, because a swollen can indicates that there's some sort of something generating gas inside, and that generation of gas could be a Clostridium culture growing.
And if the can is dented as well.
And Amanda shared that if it's dented, it's a concern as well. Okay, so what are some characteristics of this bacteria? Heat can destroy the toxin. I'm wondering if that true? I have to look at it, because I know you heat treat food, you boil it for a certain amount of time. So maybe 120 degrees is what temperature you need to get the food to in order to destroy the toxin, but not just a low heat. We'd have to look that up. I'm curious if that's right. They're anaerobic, they form spores, and it's the spores that are the concern. It's because the spores survive treatment. And if you have a food that's on the shelf and it doesn't have a lot of acid in it, and it's stable on the shelf, the bacteria can survive there. Bacteria of this nature can survive 5% salt, and they have very heat resistant spores. The spores can survive a lot of heat. And these spores are produced if the pH is above 4.8. So if you reduce the pH, then you won't get the toxins produced. Next slide. What are some risks? Anything that's canned. So vegetable, canned meat, canned corn, canned peppers, canned green beans, canned soup, canned asparagus, tuna fish, spinach, pate, lunch meat, ham, sausage. All of these foods can be a concern. Also, another concern, honey. I don't mention that there. Uncooked vegetables that have close contact with the ground, for example, potatoes, asparagus, cabbage. So because it's environmental, it's in a lot of places, that's why it's a concern. If you have smoked or salted fish, seafood, that's also a concern. Honey is also associated with infant botulism. This is why typically we do not recommend feeding honey to a baby, because babies do not have a developed immune system, and they can get an infection from honey. So stay away from feeding babies honey. Refrigerated foods with a long shelf life are also a food risk. Here's my picture that I was saying. So here's an image of a poor individual who has been using, who's a narcotics user. This is from the New England Journal of Medicine. And you can see his eyes are droopy. So he's got this kind of lack of muscle control. And he's also gotten large wounds from where he's injected a contaminated needle. So the illness is produced by what are referred to as vegetative cells. And when I say vegetative cells, do you remember what that means? Just the opposite of dormant. Vegetative means they're growing.
Oh, vegetative.
Okay,
Sorry,
so
I have plants in a vegetative state.
plants are in a vegetative state. Does that mean they're dormant?
No, when my plants are in the vegetative state, that means that they're pushing out new leaves there.
Oh, vegetative states means they're growing, actively growing. Okay.
So
Oh,
in that context.
okay, yeah. Says you have to heat above 85 degrees Celsius, according to Paul. Thank you, Paul. So you can get rid of the toxin, but you have to heat it above 85 Celsius. The Clostridium botulinum toxins, the most toxic, potent toxin that we know of, and you only need a low dose for it to be destructive. I took this from a science paper just to kind of show you an explanation of how this toxin works. So imagine, if you will, you have a neuron. A neuron, can I draw here? The neuron has a shape where it's kind of like the mushroom shape. And inside of the neuron, you have vesicles, and the vesicles are all filled with a chemical compound called acetylcholine. And what happens with the acetylcholine under normal circumstances, they have proteins attached to them. And the proteins come up to the surface of muscle maybe, and it attaches the muscle to the vesicle, and these proteins attach on both sides. And when that happens, the acetylcholine gets the vesicle gets attached to the muscle, and then it releases the acetylcholine into the muscle. So the muscle works in a normal way, is because the acetylcholine is going into the muscle. So it's the nerve is sending a message. And what happens with the case of the toxin is that normal operation is no longer working. This is shown in the picture on the right-hand side. The right-hand side shows a picture where there's the botulinum toxin attaching to these proteins, and it breaks these proteins down. So if there's a toxin attaching to the protein gets broken down. And so when that protein is broken down, the acetylcholine is lost. And so there's no message going to the muscle. So as you can see here, the acetylcholine is lost. And now toxin goes into the muscle, and the muscle just doesn't get the message. So the muscle doesn't know that it has to respond or it has to move. So we have here neurotoxin binds to the nerve endings, blocks the release of acetylcholine, chemical necessary for the transmission of nerve impulses. Okay, so that's there. If you suspect that you do have a botulinum infection, it can be fatal. So it should be considered as a emergency. So, and you can have symptoms. So the lethargy, the muscle tone loss can happen within 12 hours to 3 days. Okay, so symptoms, progressive flaccid paralysis. That means that the more infected you get, the more flaccid, the less your muscles work. You get vomiting, constipation, diarrhea. It's very similar kind of symptoms for a lot of bacteria infections. But this is bad because you also get blurred vision, double vision, drooping eyelids, slurred speech, difficulty swallowing. It's almost like somebody's having a stroke. There's also paralysis. So the muscles are working less and less and less as you go along. And it says here, infants with botulism appear lethargic, feed poorly, and are constipated, have a weak cry and poor muscle tone. So if your baby's not crying, even though it's sick, that's a serious concern. Does it cause tetanus lockjaw? I don't know. I know tetanus causes lockjaw, but I don't know if botulinum causes lockjaw. Good question. It's a question from Paul. Anybody can answer that, go ahead. If you want to look up on the internet. What is the name of the organism that causes lockjaw? Is tetani. I'm trying to remember the full name of it. Is it Clostridium tetani? Tetanus caused by, I'll carry on. That's a good question. Tetanus is what they give you an injection and vaccination for in case you step on a rusty needle, because if you have the tetanus bacteria, you can become paralyzed from that infection. Other symptoms of botulism, muscle paralysis, and if it's untreated, it can be paralyzing the body, and it can occur as early as 6 hours, or it can go as far out as 3 days to 10 days even, it says there. How do you prevent it? That was a question earlier. Low acid foods, heat to 121 degrees Celsius for 3 minutes. Paul mentioned that it is also deactivated at 85 degrees Celsius for a minute. If you do it for 80 degrees for 6 minutes. So there's lots of different temperatures you can treat for longer periods of time. The active cells for botulism can be destroyed by heating to 60 degrees Celsius. Also down at the bottom there, spores inactivated by ozone or chlorine dioxide. So if you oxidize the spores, you kill the spores. And it's heat resistant of the spores is decreased when the pH is below 5 or above 9. Okay, symptoms of botulism. Anybody? Vomiting, yes. Diarrhea, fever,
Dizziness.
dizziness, and paralysis, the important one.
Paralysis.
Yeah.
Would you say that muscle aches?
Muscle aches, yeah.
Also, loss of muscle coordination.
Loss of muscle coordination, yeah.
Muscle tone?
Also vision.
The vision, drooping of the
Yeah. Describe a method of eliminating vegetative cells. Starts in the face and works down, Paul says. Yeah.
Eating to over 85?
Heating over 85 degrees Celsius, what we just saw, is a way of eliminating vegetative cells.
acidity with the pH of less than 4.
Make it more acidic. Yes, that works as well. Why do Bacillus Clostridium toxins affect the nervous system? And that was the explanation with the acetylcholine vesicles being disrupted. So the nerves are disrupted. The regular operation of the nerves is disrupted. You see a recommendation to not feed infants honey. Why?
They don't have an immune system capable of combating botulism.
I don't know if you all heard that online. Amanda said infants will not have fully system. Okay, so that's botulism. On to our next case. And that's the case of Walkerton. So if you're older than 22, you'll remember Walkerton is a case, a study in water management. This happened in the year 2000, 22 years ago in Ontario, where 22 people lost their lives. No, sorry, six people lost their lives in this small town, and people are still sick from the E. coli infection that occurred there. Okay, so let me talk about this bacteria called E. coli 0157H7. This is enteric. Enteric means it's in the intestines. So whether it's the last part of the colon or small intestines, it's found within the gut of animals. So it's also in water, contaminated by feces. So that's where this bacteria is. I say there it's found in cattle, sheep, and deer, but it's more than that. What are some characteristics? It doesn't form a spore. It's a gram-negative rod. It can survive without oxygen, so it's anaerobic, but it can live if there is oxygen, so it's facultative anaerobic. It's not really motile. It's not known for its motility, but it can survive in frozen food. And it's the toxins that cause severe damage. So the toxins from the bacteria, again, just as in the case of the Clostridium botulinum toxin, the toxin in E. coli is called a Shiga toxin, and the Shiga toxin is what causes the damage. I have a picture here of what's called a Petri film. Here you can see blue colonies, and they have a little bubble around it. That would be your E. coli. So if you had some fecal material and you tested that for bacteria that live there, those are referred to as coliforms. They do this test at water treatment plants to find out if the water is contaminated with feces. So you have a water sample, you plate it out, and you see if there's growth, and if there's these E. coli bacteria, then you may need to treat your water more. Yeah. I think so. E. coli, I just want to share where the 157 comes from here in this picture. So there's lots of different kinds of bacteria. There are E. coli bacteria that survive. And there's a whole bunch of different blood types that have been tested against these bacteria. And they've got 200 so far. And there's this thing called EHEC. EHEC, enterohemorrhagic Escherichia coli. There is a long list of them. This long list of serotypes, they're referred to as the O serotype, is they're in 5 virulence groups. So they have different ways of being virulent. And the EHEC 157 is in this one particular group. Let me just show the next slide. So there's this thing called an O antigen, and it's a polysaccharide. When I say it's an O antigen, what they've done is they have a immunology test. And if the immunology test is positive, it has this antigen called an O antigen. There's another antigen test called an H antigen test, and the H antigen tests whether this E. coli bacteria react with the immune system. And in that case, it's the flagella. So there's the O antigen and an H antigen from E. coli that activate our immune system. Like our systems recognize these parts of the bacteria. And that's why we have these immunity tests based on them. And the O stands for a German word, own, and the O is like a polysaccharide chain off of the bacteria. There's a K antigen as well as an H antigen, and the H antigen is a German word for the flagella. So we get these O and H from the German words for flagella and polysaccharide chain. So you see here, there's the E. coli O157H7. So there's a reason for the name, is that they've done these tests to figure out where the, what parts of the immune system react to it. So where do we get it? We can get it from uncooked meat, raw milk, alfalfa sprouts, unpasteurized fruit juice, lettuce, cheese curds, salami, sausages, different ways of getting it. The food infection, I say there it's undercooked ground beef, apple cider, milk, ham, turkey, roast beef, raw vegetables, cheese, contaminated water. And if you contaminate water, it can go from person to person by hand or mouth contact as well. And here are the symptoms, common symptoms: stomach cramp, diarrhea, fever, nausea, vomiting. But in this case, the diarrhea is bloody diarrhea. So it's kind of extreme, quite extreme. How do you prevent it? You inactivate the toxin by heating it. So you have to cook hamburgers to a certain temperature, and that's why we have temperature regulations on cooked meats. Thoroughly wash your fruit and vegetable. Acidify, again, acidifying can denature the protein toxin. Here's a complicated word. Benzalkonium chloride is a food sanitizer. So it's probably a sanitizer that the food industry would spray down, whether it's food preparation surfaces, to make sure it's clean. Ethanol and hot water are effective as well. And it's also sensitive to ultraviolet radiation. So ultraviolet radiation can break down the bacteria too, and the protein. So what happened at Walkerton? Walkerton is a small town far west of here. I'm not too sure exactly the location. It's in Western Ontario, I think. So 2,000 people in this small town got sick from an E. coli infection. Some people are still sick with the results of that infection. And they found 2 organisms responsible for their sickness, E. coli O15787, as well as Campylobacter jejuni. And the investigation of where this happened was the water plant. There were 2 guys who were working the water plant. They pled guilty to not paying close enough attention to the water quality. And so because of their, they pled guilty to common nuisance from the contamination. And there's some Wikipedia sites as well as CBC video based on Walkerton outbreak. Here's a long list of some outbreaks that have occurred from 1982 to 2001. And as you can see, what kind of foods can make this happen? So we have lots of locations in the United States and Canada: hamburger, seafood, cold sandwiches, potatoes, raw milks, hamburger, frozen beef patty, turkey roll, ground beef, roast beef, patties, water, school lunches, apple cider, hamburger meat, garden salad, tabbouleh salad, cured salami, pasteurized milk. So somebody was serving pasteurized milk. And Paul's got a note here. This is what we use in the post-harvest on campus for vegetable wash. So there's a vegetable wash for cleaning your vegetables at the Food and Farm Center. Paul, is that correct? Post-harvest? Okay. Hey, make sure it's clean and sanitized. It kills E. coli and other pathogens. Yeah. The worst on this list is a white radish sprout. You see almost 9,000, 10,000 people get sick in Japan from that, and there are 3 deaths. So this list has a list of sicknesses as well as deaths. You could, I just shared that for your information. Okay. So what makes E. coli O15787 so hazardous?
Some of the symptoms maybe are pretty serious.
Very serious symptoms, especially the bloody diarrhea, because it attacks the intestines and causes lesions. And it can come from undercooked food, so it can be a common sort of infection. And I've given some sources for E. coli.
I remember when watermelons was a source of E. coli. I was telling someone in the class I've always washed my watermelon for about the past 25 years. I wash my watermelon before I cut it. I put it in the sink when I get from home and I soak them and rinse them. Because I remember hearing of someone getting sick from the cut watermelon. As you cut through the brine, you drew the bacteria into the middle of the fruit, and that's how they got sick. So I've always washed my watermelon.
I don't know if everybody's heard that. Natalie shared that she's washed watermelons for the last 25 years due to the fact that the outside of the watermelon can become contaminated, and if you cut it, the bacteria can get inside. And Don says lettuce is a source of, that's true. Thanks, Don. That's another source. Watermelons. You can think of lots of different vegetables. You've heard of different recalls that have occurred due to the E. coli as well.
I think that the hydroponic growing of lettuce, like lately, because they do a lot of lettuce.
Hydroponic of
hydroponically and
lettuce,
vertically, that
yeah.
significantly reduced E. coli contamination versus when they were growing them on the ground, where you're gonna get those fecal contaminations or whatever. You're not really getting that in,
You
for
know,
example, in our leafy green setup, there's really no opportunity for E. coli because it's just sort of a sterile environment.
right.
It's
So
supposed to be.
Natalie is sharing that your leafy green environment doesn't have the E. coli.
The water that comes there is not treated water.
No,
So
well, the
we
water
can-
would be, I guess, that.
No, if you treat, like, you cannot put chlorine in the water used to grow vegetables. It will kill the vegetables. So it might not be treated, so there is also a risk there.
Yeah.
Using untreated water is a risk with
very cool.
vegetables, yeah. Because
That would be-
you want treated water or some sort of cleaned water, filtered water.
There
Yeah.
was a time I had an experience. Water at almost 79 liters deep. Used to, it was pumped out. There was sample of E. coli. E.
Yes.
coli, the water is 70 liters deep.
And Tabachku shared that there was a sample of water from 80 meters down, so a very deep well, and the deep well still had E. coli in it. Yeah, they can survive lots of places, hey? Yeah. And 2000, what was the suspected source of contamination? Manure of cattle used to fertilize nearby crops. Thanks, Don. Yeah. I think that's what it was. Natalie's got a
Did you say it was E. coli is an aerobic?
facultative anaerobe. So it can survive aerobically, but it is normally anaerobic. It can live both places. Okay, so that's the E. coli 157. Staph aureus. I just mentioned Staph aureus here because Staph aureus is on our skin. It's a common sort of microbe. It's found in hair, nasal tract, so it's in our noses, our oral passages, acne, it's on fresh water, plant surfaces. It's a very common bacteria. It's a,
People have a staph infection.
yes, if you have a staphylococcus, Staph aureus infection, that's what is caused. Can be a common bacteria, but there are some bacteria that just cause infections. So most don't, but the occasional one does. It can be a very nasty skin infection.
I had a staph infection in the hip bone.
In the hip bone, oh my goodness.
I spent a month in the hospital when I was young.
That sounds painful. Do you know if they treated with antibiotics?
Oh, yes.
Yeah, antibiotics, lots. So Staph aureus, facultative anaerobe. So it can survive again anaerobically, but it's also with oxygen too. It's gram-positive, round shape.
Is that foodborne?
Not necessarily foodborne, unless it's the skin of the animals.
Oh, well, yeah.
It's resistant to salt, so you can have salt go up to 20%. It's heat resistant and it produces toxin. The bacteria themselves are killed by mild heat, but the toxins are very stable and you can't deactivate them until you get to like 121 degrees Celsius. So it's a real severe heat treatment. You need to push the Staph aureus bacteria to remove the toxins. So staph infections can occur in meat and food products, poultry, eggs, salads, potato, macaroni, anything with a cream-filled filling, cream pies, chocolate eclairs, milk products, and any food that requires lots of handling. So you have food risks include poultry, cooked meats, raw milk, especially hard cheeses, salad, and cold sweets. They're a fast-acting toxin, and they can affect you in as little as 30 minutes. So you could eat it, and half an hour, you can have your nausea, vomiting, stomach cramp, diarrhea. And usually the infection is mild. And if you have a staph food infection, it's not contagious. You can't transmit the toxin from one person to another. It's the skin that you want to wash off. So if you have food fermentation, lactic acid bacteria can produce something that can inhibit Staph aureus. So milk fermentation is a lactic acid fermentation. So if you have yogurt, buttermilk, what other, kefir, that sort of product can inhibit the Staph aureus. We have chemical sanitizers like quaternonium. If you reduce the pH of your food to 4.2, a lot of these things are if you reduce the pH. And that was it for Staph aureus. So next topic in this long section is sterilization. Paul's got the chemical name for ingredients for the vegetable wash. So lactic acid is there, sodium bisulfate is there, and sulfonic acid. So it really acidifies it with two different kinds of acid. And sodium bisulfate, I wonder what that does. But that helps with the removal. And lactic acid as well, 30%. Thanks, Paul. Okay, so carrying on. I know the next section was biological agents in food, but I'm going to move to sterilization. And here's a picture of some very large autoclaves. These autoclaves are huge. What you do is you have a forklift. The forklift puts the food items into these autoclaves. This is how large they are. Sterilization, the word sterilization means eliminating all forms of life, eliminating or killing it. So that's a sterilization. And what we're doing is removing fungi, bacteria, or viruses when we're sterilizing. Sterilization's on a surface. So when I ask you to use a sterilization liquid, I'm asking you to sterilize the surface with some sort of compound. You can sterilize the top of a bench. You can sterilize fluid. You can also sterilize culture media, right? So there's something that we do in lab is we cook the media in an autoclave, and that sterilizes the media. So you can sterilize with heat. You can sterilize with chemicals. There's also irradiation. Irradiation exposes your food to a radiation source, and high pressure could also kill off these organisms. And you could also sterilize with filtration. So there's a number of ways you can sterilize something. And I've got some YouTube videos for botulism, to avoid botulism, for canning food, and how an autoclave works. I've also been asked the question, like, how do you sterilize equipment in a hospital? So I found this video on sterilizing hospital equipment. So if you want to see how they do it in a hospital. This is a great video. The woman shows a tour of the hospital and how they sterilize all the medical equipment, whether it's the sterile tweezers, sterile scalpels, sterile, like you can imagine in a hospital, you have operations, all that equipment needs to be sterilized. So this is a video on that. So that's sterility. Next word, disinfection. So a disinfection is a process that removes some of the contaminants or kills some of them. And this can be for anything like fungus, bacteria, viruses, or spores. And it reduces the live cells in a number of ways by treatment. And again, disinfection, you can disinfect a surface, you can disinfect a fluid, you can disinfect your culture media, and you can do this by heat, chemicals, or irradiation. There's 3 different ways of disinfection. And here's an example of disinfection. Most of you have had this in your homes or have seen a bottle of this. I've taken a picture from London Drugs. Just what is on Mr. Clean. You take a look at the bottle. It says green liquid. What's in the bottle? Active ingredient, sodium hydroxide. So NaOH is the active ingredient. And if you take a look, it's 0.434%. So to make a 0.4% solution, you need 0.4 grams of sodium hydroxide into 100 mils. So there you've made Mr. Clean. Yeah, that's one way. There's other ways of making Mr. Clean as well. We have another group coming here in 7 minutes. Oh, this is new. Thanks. Okay, so some treatments include acidal treatment. Acidal means that it kills the microbes. Biocide, fungicide, germicide, virucide, sporicide. So all of those are side, they kill all those things. And there's also static. So it reduces the numbers, inhibits the growth maybe, but it doesn't really kill them. So if something is bacteriostatic, it's referred to as reducing the life of that organism. So for example, refrigeration can be bacteriostatic or fungistatic. It can reduce the fungus, it can reduce the bacteria. And final slide, what is sterilization? What is disinfection? Explain how the autoclave oven works. I'll cover that in class next day. We'll talk a little bit more about the autoclave oven. What is the difference between static and acidal? Okay, so I'll leave that to you guys. We just looked at those things. What is sterilization? What is disinfection? I want to talk about tomorrow's lab briefly. Tomorrow in lab, you do a follow-up. If you remember before break, I asked you to make 3 things. Kombucha, yogurt, kimchi. So one of those 3 things, you and a lab partner both will come in, divvy it up between the 2 of you. So one person do one food-based organism, the other do another food-based. If you're on your own, just do one. And each of you will do a gram stain of what's in there and set up a microscope so that each of you can kind of swap the slides, take a look at what's in there, and you'll do a follow-up, draw the picture of what kind of bacteria there are, and tell me if they're gram positive or gram negative. That's the first thing. Then there's the experiment for tomorrow. We're going to do isolation of bacteria. The isolation of bacteria is you take some bacteria from a source, spread that out onto a plate, cross-streak the plate, There's also the pour plate method where you take the bacteria from a source, pour it into a plate, and then add media to it, and then let that grow for a week. And then the next week, we'll see how your cross-streaking worked. We'll see how your pour plate works. So you have a follow-up to last week's experiment tomorrow morning, and then you'll do the experiment. And then that experiment, you'll do a follow-up to that one next week, okay?
We'll start our plates.
You'll start your plates tomorrow. Yeah, okay. Thanks for listening to me, everybody online. I'm gonna stay online in case you have another question or something. But for now, thanks. Oh, and if you're staying here, oh, we can't stay in this room. We have other people banging on the door almost. Okay. Singing in the hallway. Thanks, everyone. See everyone tomorrow in lab. Thanks, Cass. Thanks, Will. See you tomorrow. Israel, see you tomorrow. And Danielle says see you tomorrow. Yep. Paul. Cheers. See everyone tomorrow. I'm going to sign off now. Like I said, I got a class coming in.
Okay, bye.
See you tomorrow morning, bright and early.
Bye, Toby.
If anybody has a question for me online, go ahead and email me. Talk to you tomorrow. Cheers.